what if the secret to healing, the most impossible, like radiation-scorched wounds in the human body, isn't giving those cells what they need to survive, but actually intentionally suffocating them?
Yeah, it feels completely paradoxical when you first hear the concept because we are just so conditioned to think of oxygen as the ultimate driver of metabolic health, right?
Right.
And tissue repair.
Right.
But when you actually look at the raw data, inducing the sort of controlled state of physiological panic, it actually forces biological systems to produce a profoundly more potent healing response.
And that biological panic, that's really the core of our deep dive today. We are exploring a major paradigm shift in regenerative medicine, specifically how the secretome profile of hypoxia-preconditioned stem cell exosomes,
Sorry, a mouthful, yeah.
it is, but how that profile enhances microvascular repair in severely irradiated or, scarred tissue.
Mm.
And for you listening, we're pulling from a really fascinating stack of cutting-edge biomedical research today.
Yeah.
This is anchored by a pivotal PLOS-1 study on radiation-induced salivary gland damage, alongside some wild new frontiers in cell-free therapy and nanotechnology.
Yeah, and for anyone tuning in who tracks the evolution of regenerative medicine, you definitely know the frustrations surrounding these biological dead zones.
Oh, absolutely.
Because tissues subjected to severe radiation therapy or, chronic ischemic scarring, like advanced diabetic ulcers, they simply lose their native capacity to regenerate. I mean, the underlying cellular infrastructure is effectively just annihilated.
Right. I was reading through the clinical realities of those dead zones in our sources, and it's not just that the surface tissue is damaged, right?
Yeah.
The entire supply chain is gone. And for decades, the holy grail for fixing this was traditional mesenchymal stem cell therapy, or MSCs. The whole logic was to just take live, cultured stem cells, inject them into the patient, and wait for them to engraft and physically differentiate into new tissue. But the literature shows a much messier reality.
A significantly messier reality, and one that is, frankly, fraught with systemic bottlenecks. I mean, when you administer whole MSCs intravenously, the first major hurdle you hit is the pulmonary first pass effect.
Right.
A massive percentage of those live cells simply get trapped in the capillary beds of the lungs. They never even reach the ischemic target site.
Wait, so they just get stuck in the lungs. They don't even make it to the injury.
A huge portion of them, yeah. And if a fraction of them do manage to reach the target, they're dropping into a highly inflamed, incredibly hostile microenvironment. The survival rates of cultured MSCs in ischemic tissue are notoriously poor.
Yeah, they just die off.
Right. Plus, there's the inherent risk profile of transplanting live, self-renewing entities. The sources we're looking at explicitly note the documented risks of unwanted malignant tumor formation.
Wow.
Or even ectopic tissue growth when you introduce cultured bone marrow MSCs into this dynamic, damaged environment.
Which is, that risk profile is exactly why the field began shifting its focus, right? Because researchers observed that patients were still experiencing therapeutic benefits from MSC treatments, even when the injected cells were dying off rapidly.
Yes, even when they were failing to engraft.
Which led to this realization that MSCs, they act less like replacement parts and more like localized dispatch centers.
Exactly. It's called the paracrine effect. The MSCs essentially evaluate the inflammatory environment, and they begin secreting this highly customized cocktail of trophic factors, cytokines, and genetic material to orchestrate the repair process.
So the cells themselves don't become the new tissue.
No, not at all. Their secretome provides the instructions for the surrounding surviving cells to actually do the work.
So the secretome is the true therapeutic engine here.
It is.
And the primary delivery vehicles for this secretome are exosomes, right? These tiny lipid bilayer nanovesicles. And we're talking typically ranging from, 30 to 150 nanometers in diameter?
Yeah, incredibly small. And because they're just a fraction of the size of a whole cell, they easily bypass that pulmonary entrapment we talked about. They just slip right through and penetrate deep into ischemic tissues.
It fundamentally changes how we visualize the therapy, I think. Like, instead of sending an entire team of vulnerable architects and construction workers into a toxic disaster zone,
where they probably won't survive, yeah.
Right, where they might die, the stem cells instead dispatch a fleet of automated drones. And these exosome drones, they aren't just dropping off passive supplies. They're delivering an active override code, specifically proteins and microRNAs, right into the cytoplasm of the local surviving cells.
That concept of the override code is so vital to understand. We are talking about delivering messenger molecules that actively silence self-destruct sequences and force those recipient cells to rebuild.
Right.
But here is where the methodology in our sources takes a really fascinating turn, because researchers aren't just harvesting these exosomes from stem cells grown in, standard, comfortable laboratory
Yeah.
conditions.
Right. They're not pampering them.
No, they are engineering the cargo by intentionally starving the parent cells of oxygen first.
Hypoxic-preconditioning. Let's really break down the mechanics of this because I think it's so cool. Standard lab cultures incubate cells in about 21% oxygen, which is normoxia.
Correct.
But inside the human body, tissues operate at much lower oxygen tensions. And inside a biological dead zone, like an irradiated tumor bed, oxygen levels just plummet drastically, often hovering between, 1 and 5%.
Yeah, very low. So when you culture MSCs in 21% oxygen, they are essentially living in a state of unnatural luxury.
Like a spa.
Exactly. They have zero biological incentive to produce an aggressive crisis-level secretome. But by moving them into a hypoxic chamber and dropping the oxygen down to that 1 to 5% range for, say, 24 to 48 hours, researchers simulate the pathological environment of a severe injury.
Okay, but I want to challenge the biological logic of this for a moment. Because if you take a cell out of its comfortable 21% oxygen environment and you subject it to a suffocating 1% oxygen environment, doesn't cellular metabolism just begin to shut down?
You would think so, yeah.
Like, why does starving the cell actually improve the therapeutic density of its exosomes instead of just killing it?
It comes down to a highly conserved evolutionary survival mechanism. And this is primarily mediated by a master transcription factor called HIF1 alpha. That's hypoxia-inducible factor 1 alpha.
Okay.
Under normoxic conditions, HIF1 alpha is constantly synthesized by the cell, but then it's rapidly degraded. It's essentially kept in check. But the moment oxygen levels drop to that 1 to 5% threshold, that degradation process just halts completely.
So the HIF1 alpha stabilizes. It accumulates in the cytoplasm, and then it translocates into the nucleus to act as basically the ultimate panic button.
Yes. It binds to specific DNA sequences and completely rewires the cell's genetic expression profile.
And that rewiring triggers a massive triage response.
Right. The cell recognizes it's in an ischemic crisis, and its primary directive becomes vascular recruitment. It needs blood flow immediately.
Oh, yeah.
So consequently, the secretome it packages into its exosomes becomes hyperloaded with pro-angiogenic and anti-apoptotic factors.
Meaning it builds blood vessels and stops cell death.
Exactly.
The literature specifically highlights massive upregulation of VEGF, which is vascular endothelial growth factor, and GM-CSF in this hypoxic secretome. But going back to that override code we discussed earlier, it's the non-coding RNAs that really drive the transformation, isn't it?
Oh, absolutely.
Because the sources point to a massive enrichment of specific microRNAs, particularly miR210, which is known in the field as the hypoxemia genetic signature.
Yeah, miR210 is intimately tied to the hypoxic response, right alongside miR126. And I think it's really important for us to understand how these microRNAs function once they are actually delivered by the exosome.
Walk us through it.
Well, they don't just float around randomly. They engage in epigenetic silencing. So they bind to complementary sequences on target messenger RNAs inside the recipient cell, and they physically prevent the translation of specific proteins.
Okay, so they act as biological brakes.
Exactly. Biological brakes.
Like if a cell in an irradiated dead zone is trying to translate proteins that will trigger apoptosis, program cell death, the microRNAs delivered by the exosomes bind to those transcripts and just silence them. The cell is effectively blocked from dying.
And that combination, silencing cell death while simultaneously delivering high concentrations of VEGF to stimulate growth, that is what makes the hypoxia-preconditioned secretome so unbelievably potent.
The 1-2 punch.
It really is. But to truly appreciate its efficacy, we have to look at the primary target of these exosomes in the damaged tissue.
Right, the microvasculature. Because when you look at chronic scarring or tissue exposed to heavy radiation, the fundamental reason the tissue fails to heal isn't just the loss of the surface epithelial cells. No, it goes much deeper. The radiation causes massive DNA double-strand breaks in the endothelial cells. Those are the cells lining the blood vessels, right?
All right.
So those cells enter senescence, the vessels collapse, and the entire microscopic plumbing network is just obliterated.
And chronic ischemia is the ultimate roadblock to regeneration. I mean, you cannot rebuild complex tissue without a functional microvascular network supplying oxygen and clearing out metabolic waste.
Right, it's like arriving at a burned-down house. You can't just put up new drywall. You have to rebuild the tiny, intricate plumbing inside the walls first, or the house will never function.
That's a perfect analogy. Therefore, the primary objective of these preconditioned exosomes is to target those surviving damaged endothelial cells and force them to reestablish the vascular bed.
So I was looking closely at how the exosomes interact with HUVECs, that's human umbilical vein endothelial cells, which are often used in these lab models to study vascular repair.
Right.
Standard lab model.
And when the hypoxic exosomes are introduced, the endothelial cells endocytose them, like they literally engulf the nanovesicles. And then the exosomes release their payload of miR-126, miR-210, and VEGF directly into the cytosol.
And once that payload is released, it initiates this profound cascade of survival signaling. Most notably, it activates the PI3-catenose pathway.
Okay, let's trace that actual cascade rather than just, treating it as an alphabet soup of acronyms.
Sure.
When the exosome cargo triggers this pathway, PI3K gets activated at the inner cell membrane, which then recruits and phosphorylates AKT.
Right.
And once AKT is activated, it acts as a central kinase, regulating a multitude of downstream targets.
Crucially, phosphorylated AKT activates ENOS, that's endothelial nitric oxide syntose.
And this enzyme immediately begins synthesizing nitric oxide, which is a potent vasodilator and just a critical signaling molecule for endothelial proliferation. Furthermore, the activation of this pathway drastically suppresses the accumulation of reactive oxygen species, or ROS.
And that ROS suppression is vital, right? Because irradiated tissue is basically drowning in oxidative stress.
Drowning in it, yes.
So by neutralizing the ROS and generating nitric oxide, the endothelial cells are pulled back from the brink of apoptosis. They stop dying, and they transition into a proliferative state.
Which leads to this incredible phenomenon of tube formation. In vitro, when you apply these hypoxia-preconditioned exosomes to damaged endothelial cells, you can literally watch them under a microscope stretch out, migrate toward one another, and self-assemble into three-dimensional capillary-like networks.
Oh, wow.
They are physically sprouting new microvessels.
Relaying the microscopic plumbing so the dead zone can finally breathe. It's such an incredibly elegant cellular mechanism. But what makes our sources today so compelling is that they don't just leave this in a petri dish, right? The PLOS-1 study provides a rigorous in vivo application of this exact mechanism, addressing a devastating clinical challenge.
Right. The researchers targeted radiation-induced xerostomia.
Dry mouth.
Severe dry mouth, yes. For patients undergoing radiation therapy for head and neck cancers, the collateral damage to the salivary glands is just profound. The radiation destroys the acinar cells, the microvasculature, and the resident stem cell populations,
leaving the patient with this severe, irreversible condition. And current standard of care treatments are purely palliative, right? Just artificial saliva substitutes or muscarinic agonists that try to squeeze a little more output from whatever functional tissue remains.
Exactly. Standard medicine basically considers this biological dead zone permanent.
But the PLOS-1 methodology approached this by leveraging that hypoxia hack we've been talking about.
They did.
They cultured human adipose-derived MSCs, so stem cells isolated from human fat, and they placed them in a hypoxic chamber to trigger that HIF-1 alpha cascade.
Right.
They then collected the conditioned media and used ultrafiltration to isolate the high molecular weight fraction of the secretome, which is highly enriched with those exosomes and protective proteins.
And what's fascinating is the delivery method here. They took mice that had received 15 grays of localized radiation to the neck, which is a massive dose, designed to obliterate the salivary glands, and they administered this secretome intravenously via the tail vein.
Wait, so they didn't inject it directly into the neck?
No.
Yeah.
They relied on the exosomes to navigate the systemic circulation and home in on the site of severe inflammation.
That's wild. Systemic circulation.
Yeah. The homing capability of exosomes to sites of tissue injury is a massive advantage of their lipid bilayer structure and surface receptor profile. And the physiological rescue observed in these mice was just remarkable.
I bet.
Compared to the control groups, the mice treated with the hypoxic secretome showed significantly improved body weight recovery and a dramatic restoration of salivation capacity.
Yeah, I saw the researchers measured the actual output, noting severe increases in salivary amylase and epidermal growth factor, or EGF, in the saliva of the treated mice. But the most compelling evidence to me comes from the histological analysis.
The tissue slices.
Right. They looked at the tissue under a microscope to see if that PI3-catenose pathway had actually done its job in vivo.
And the histological data confirmed that the secretome physically preserved the cellular ecosystem of the gland. It wasn't just a temporary functional boost. It was real structural protection against radiation-induced senescence and apoptosis.
Let's look at the specific cellular markers they quantified, because this is where the rubber meets the road. They saw significant preservation of CD31, which directly confirmed the protection of those endothelial cells and the microvascular network we've been discussing. They also found preserved expression of AQP5, marking the functional acinar epithelial cells, and alpha SMA for the myoepithelial cells that actually contract to secrete the saliva.
And perhaps most importantly, they noted the preservation of c-KIT expression, which is a key marker for resident progenitor cells. So by preserving the microvasculature and the local stem cell niche, the exosomes allowed the gland to maintain its own intrinsic capacity for long-term self-renewal.
Which perfectly validates the shift from whole cell engraftment to exosome-mediated repair. I mean, the MSCs were never needed in the tissue itself. Their hypoxic care packages were enough to shield the entire salivary ecosystem from 15 gray of radiation.
It's incredible. But this brings us to a critical operational challenge that's highlighted in the literature, which is manufacturing scale.
Ah, right.
While the efficacy of hypoxia-preconditioned exosomes is clear, isolating sufficient therapeutic quantities from standard 2D flat laboratory dishes is incredibly inefficient.
It just doesn't yield enough.
No. If this is going to transition into a scalable clinical reality, we need massive yields.
And our sources point toward 3D spheroid cultures as the definitive solution to that bottleneck, right? Instead of growing the stem cells flat on a polystyrene dish, researchers are forcing them to aggregate and grow as three-dimensional spheres.
Yeah, and culturing cells in 3D spheroids fundamentally alters their mechanotransduction. It basically mimics the spatial constraints and high cell-to-cell contact found in actual human tissue.
Makes sense.
And this alters cytoskeletal tension, which naturally stimulates a much higher rate of vesicle shedding.
And when you combine that 3D spheroid architecture with the hypoxic preconditioning, you get a synergistic amplification. I mean, the cells produce drastically higher quantities of exosomes, and the cargo profile, the density of miR210, miR126, and VEGF is exponentially enriched compared to standard 2D hypoxic cultures.
Exponentially.
It solves the manufacturing yield while simultaneously boosting the therapeutic potency.
Exactly. And it paves the way for what we now categorize as cell-free therapy. The implications for clinical logistics here are massive. Whole stem cells require complex cryopreservation, careful thawing protocols, and stringent HLA matching to avoid immune rejection.
Right, they're fragile.
Very, and they have a notoriously short shelf life once prepared.
But because exosomes lack a nucleus and have a very low profile of major histocompatibility complex molecules, they exhibit incredibly low immunogenicity. Like, your immune system doesn't flag them as foreign invaders the way it would with unmatched live cells.
Precisely. They are stable, they're easily lyophilized or freeze-dried, and they can be stored in standard medical freezers for extended periods. We are really looking at a future where a trauma center or a burn unit could stock standardized, off-the-shelf vials of regenerative exosomes ready to be reconstituted and administered intravenously at a moment's notice.
It is a stunning evolution in how we approach healing. I mean, we began this deep dive looking at the limitations and risks of transplanting whole, fragile stem cells into dead zones. We explored how the true regenerative power resides in the paracrine effect, those secreted exosome drones carrying override codes.
Right.
And we dissected the mechanism of hypoxic preconditioning, seeing how starving a cell of oxygen triggers the HIF-1 alpha master switch, flooding the secretome with miR210, miR126, and VEGF.
And we tracked that mechanism all the way down to the activation of the PI3 kictinose pathway, witnessing how it forces endothelial cells to suppress oxidative stress and physically sprout new microvasculature.
Yeah.
Ultimately, we saw this process successfully rescue irradiated salivary glands from permanent destruction, proving that the biological dead zone can actually be reclaimed.
The mastery of the body's own biological signaling here is just undeniable. But, unpacking all these exact mechanisms leaves me with one highly provocative thought to close out this deep dive for you listening.
Oh, where does this lead us next?
Well, think about the trajectory of the science. Yeah. We spent this entire time talking about how to optimize the stem cell, right? Growing it in 3D spheroids, suffocating it in hypoxic chambers, just to coax it into producing the perfect ratio of miRNAs and proteins.
Sure.
But if we now know the exact molecular formula, if we know that miR210, miR126, and VEGF are the active ingredients that trigger microvascular repair, how long until we remove the stem cell from the equation entirely?
A shift from biologics to synthetic biology. You're suggesting we abandon cell culture altogether.
Exactly. Imagine a scenario where a patient presents with severe radiation damage. A computer analyzes the specific ischemic profile of their tissue. We don't wait weeks to culture stem cells in a hypoxic chamber. Instead, a laboratory artificially synthesizes lipid bilayer nanovesicles and loads them with a perfectly customized computer-printed secretome.
Wow.
The exact concentrations of microRNAs and recombinant proteins required to activate that patient's specific endothelial pathways.
A fully synthetic, bespoke regenerative drone.
Yes.
I mean, it would completely eliminate biological variability and scale infinitely. It is the logical, albeit mind-bending, conclusion of cell-free therapy.
It radically redefines our understanding of medicine. The next time you suffer a standard injury, your body's native supply chain will likely handle the repairs just fine. But for the unhealable wounds, those biological dead zones, well, the cure of the future might not involve biology at all. It might just be a synthetic care package engineered from the ground up and dropped exactly where it's needed most.